Good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET WORLD. So yes, here we are in our third session from the chapter biological classification. So far we have completed what beta? Kingdom systems of classification, kingdom Monera and kingdom Protista and the questions related to that. And today we are going to deal with the kingdom fungi, from which every year you will get a question for sure. Kingdom fungi is the major important domain of this chapter, I am telling you. That important it is. So yes, let us get started with the kingdom fungi, after which we will look into virus, viroids, prions and the lichens. So yes, look into the general characteristics of kingdom fungi. How can you describe the organisms belonging to the kingdom fungi? They are eukaryotic, thalloid, achlorophyllous, means those which do not contain chlorophyll, and purely heterotrophic organisms. Okay, they are eukaryotic, thalloid, achlorophyllous and heterotrophic organisms. They exhibit greatest diversity in morphology and habitat. In case if the question is asked, great diversity is shown in the mode of nutrition, then your answer should be the kingdom Monera. Great diversity in the morphology and habitat, then your answer should be kingdom fungi. Okay, and here the mode of nutrition is purely heterotrophic. There is no diversity in mode of nutrition. Diversity you will see in morphology and habitat of fungi. So here in kingdom fungi you can observe the fungal species from unicellular to multicellular forms. Unicellular is like Saccharomyces cerevisiae to multicellular complex edible mushrooms and toadstools. And when it comes to the distribution of fungi, they are cosmopolitan in distribution. They exist mostly in warm and humid places. And here hyphae, if you see how fungal body is made up of hyphal strands, and group of hyphae is called as what? Mycelium. Okay, mycelium hyphal network will form what? Mycelium. How can you describe hyphae? As filamentous, long, slender, thread-like structures. Hyphae are filamentous, long, slender, thread-like structures which form network, forming up what? Mycelium. Mycelium can be of two types, aseptate mycelium and septate mycelium. Aseptate mycelium, here you can see aseptate are cenocytic hyphae, which means there is no septa which is separating the hyphal strands, which shows multinucleated or syncytium conditions. Okay, tubes are showing multinucleated or syncytium condition, which is cenocytic hyphae. Continuous tubes are observed without any septal separation, and this type of hyphae is called as primitive hyphae, aseptate hyphae, which is observed in the class Phycomycetes. Second is what? Septate hyphae. Septate hyphae will have cross walls in between them. They have cross walls in between them, and there is no multinucleated or cenocytic condition. Single nucleus is present in each tube. For example, if you see Actinomycetes, Actinomycetes will have what type of septa? Pore-like septa. Okay, Actinomycetes will have pore-like septa. Basidiomycetes will have dollipore septa. Basidiomycetes will have dollipore septa. Fine, right? This is the characteristic feature of the Basidiomycetes and Ascomycetes. You have to remember. Ascomycetes will have pore-like septa, Basidiomycetes dollipore septa, and Deuteromycetes is also septate mycelium. This is about mycelium separation, aseptate and septate. Next, if you see cell wall is made up of chitin, chitinaceous cell wall, which is also called as fungal cellulose. It is composed of what ma? Chitin and polysaccharides. Composition is very important here. Next is nutrition. Nutrition is purely heterotrophic mode of nutrition. Heterotrophic mode of nutrition, wherein heterotrophic mode of nutrition can be saprophytic, parasitic and symbiotic. Mostly it is saprophytic. Mostly fungal nutrition will be saprophytic. And in case if you see symbiotic in association with algae, it forms what ma? Lichens. Okay, wherein fungal component in lichens is termed as what? Mycobiont. Algal component is phycobiont. Okay, this forms what? Lichens, which are a very good pollution indicators. Next, fungal association with roots of higher plants. Okay, with roots of higher plants, this is called as what? Mycorrhizal roots. Mycorrhizal roots. Fine. So, fungal association with the roots of higher plants, these are what? Mycorrhizal roots. They will increase the surface area of the roots and they help in the absorption of water and minerals. Next is what? Reserve food. Reserve food material includes what? Glycogen and oil. Glycogen and oil are the reserve food material in the fungi. These are the general characteristics. Now, when it comes to reproduction, it is very important every year or every alternate year in the EAPCET paper you can check. You will get a question on asexual spores and sexual spores even in the NEET examination. That important it is. Okay, asexual spores and sexual spores of fungi. In connection with living world also they can ask you asexual spores and sexual spores of fungi, because fungal mode of reproduction will be through mostly spore formation. Here if you see vegetative mode of reproduction, it can be fragmentation, fission and budding. Fragmentation, fission and budding, these are the vegetative modes of reproduction. Asexual reproduction is by asexual spores like conidia, sporangiospores and zoospores. Amongst this, which is the only exogenously released spore, okay, exogenously produced spores, asexual spores are conidia, okay, these are exogenously released, okay, exogenously produced, exogenously produced. Rest all the asexual spores are endogenously released. Next, sexual spores. Sexual spores include oospores, ascospores and basidiospores, of which basidiospores are exogenously released sexual spores. Exogenously released sexual spores. Fine. And these are again endogenously released. Basidiospores are exogenously released sexual spores for the class Basidiomycetes, and ascospores are endogenously released sexual spores of the class Ascomycetes. These are the list of asexual and sexual spores you have to remember for sure. Next. So, this is the vegetative mode of reproduction. Of course, much explanation is not required for fragmentation, where the parental hyphae is fragmented into small fragmented hyphae and they will be growing into new hyphal network forming a mycelium. Next, budding, the parental cell above which the daughter cells are arised as buds, and these buds when detached from the parental hyphae or parental mycelium, they generate into a new fungal body. Next is what? Binary fission. Binary fission is taking place mostly by unicellular fungi, wherein a single cell divides into two by amitosis without the involvement of any spindle fiber apparatus or spindle apparatus. So, this is the basic or brief description about vegetative reproduction in fungi: fragmentation, budding and fission. Okay, fragmentation, budding and fission. Okay, next, asexual and sexual reproduction. Before getting into this, you should have a clear idea on the life cycle of fungi. Okay, as I told you, fungal body is haploid beta. Okay, fungal body is haploid. Fine. So, the haploid fungal body mycelium is haploid, from which the spores which are produced, asexual spores, are haploid spores. Asexual spores can be aplanospores, conidia, zoospores, chlamydiospores. These are what? Asexual spores. Asexual spores landing onto the suitable substrata, they will develop into a new fungal body as such. Okay, so mycelium will produce asexual spores. Asexual spores will divide by mitosis. So, as the mode of division is mitosis, there will not be any change in ploidy, there will not be any change in chromosomal number. So, the spores are also haploid. Spores on germination, landing onto the suitable substratum, they will germinate, forming a new matured mycelium body. Now, mycelium, if it is undergoing sexual reproduction follows three stages. One is plasmogamy. second will be karyogamy, and third is meiosis. So here you see beta, first haploid fungal body, of course it will produce haploid spores, yes or no, but then here in sexual reproduction gamete fusion takes place, hyphal strands will get fused, two different strains of hyphal strands will get fused. First, that step is called as what? Plasmogamy. The plasma of those two tubes will be exchanged first, and then after plasmogamy it is karyogamy. In the advanced class of fungi like Ascomycetes and Basidiomycetes, dikaryon stage is observed. Okay, dikaryon stage is observed after plasmogamy, that is N plus N condition, that is after fusion of plasma, cytoplasm, the nuclei will get fused, before which dikaryon stage is observed. Just a second. Just give a moment ma. First is plasmogamy and then karyogamy, after which meiosis will take place. Just a moment. Yeah. So here fungal body is what? Haploid. Through the asexual mode of reproduction, just a second, we need to download this. Through the asexual mode of reproduction, what is happening ma? Haploid mycelium, haploid fungal body will produce asexual spores which are haploid, they will replicate through mitosis, and then these asexual spores landing on the suitable substratum, they will give rise to again haploid fungal body. When the fungal mode of reproduction is sexual, in sexual mode of reproduction what happens? Gametic fusion takes place. Okay, what happens ma? Gametic fusion takes place. So after gametic fusion, what happens? Two different strains of fungal body will get fused. First step of sexual reproduction. In sexual reproduction, first stage is plasmogamy. In plasmogamy, what happens? Two different strains of fungi will get fused. Let us assume plus and minus strains, be it by somatogamy or gametic fusion, two different strains of bacteria will get fused, and dikaryon stage is observed in higher classes of fungi. Okay, dikaryon stage, which is N plus N condition, that is prior to karyogamy. Okay, karyogamy, which is nuclear fusion. Okay, after karyogamy, diploid stage is occurred, like that can be assumed as zygote. If it is in Ascomycetes, ascus is called, we call it as ascus, that diploid stage, and if it is in Basidiomycetes, it is basidium. Fine. These are the sites of meiosis. Sites of meiosis, which is the third stage of sexual reproduction. Okay. So, from the diploid ascus and basidium, meiosis occurs and this will produce haploid spores. If it is from the ascus, ascospores are produced after meiosis. If it is in basidium, haploid basidiospores are produced. Okay. This ascospores and basidiospores landing onto the suitable substratum, and after germination they will develop into a new haploid fungal body. Once again, for your quick understanding, I will revise, listen ma. Sexual mode of reproduction happens in 3 modes, happens in 3 steps. First is plasmogamy, second karyogamy, and third is meiosis. What is plasmogamy? 2 different strains of fungal hyphae will get fused. Let us assume it as plus and minus strain. It can be through somatogamy or it can be through gametic fusion as such. After plasmogamy, when plasma is exchanged in between 2 different hyphal strains, what happens is, before karyogamy, 2 nuclei occurs in the same tube. Okay, one septa is covering the 2 nuclei, which is giving you dikaryon structure, that is N plus N condition, dikaryon stage. After which karyogamy happens, that is nuclear fusion, and post karyogamy, this diploid cell which is formed or the zygote which is formed, we call it as ascus in ascomycetes, basidium in basidiomycetes, and these are the sites of meiosis. Okay. In ascus, post meiosis, what are formed beta? 4 ascospores from one inside one cell in ascus will form 4 ascospores, and basidium will be resulting 4 basidiospores. They are what? Haploid. After meiosis, haploid ascospores and basidiospores are formed. On germination, they will give rise to new fungal body. Okay, new fungal mycelium. This is the life cycle of what? Fungi, both asexual and sexual. Okay. If you see, most of the stages in the fungi, they are haploid stages. So, we can assume fungi is having what? Haplontic life cycle, same like that of algae. Haplontic life cycle. I hope this is clear, right? Asexual and sexual stages. Now, you see ma. So, asexual and sexual reproduction. Asexual reproduction is through spores, I told you. Zoospores are motile and biflagellate over here. It is very important for you people to remember whether the spores are motile or not, whether they are exogenously or endogenously produced. Very, very important beta. N number of times asked question. You can take a screenshot of this slide as well. So, zoospores, these are motile and biflagellate. Conidia is the only exogenously released asexual spore. Remember, Conidia is a non-motile, only exogenously released asexual spore. Asexual spores like Conidia are observed both in Ascomycetes class and Basidiomycetes and Deuteromycetes class. Deuteromycetes. Fine. Zoospores you can observe it in Phycomycetes. Next is sporangiospores. Sporangiospores are again thin-walled, non-motile, endogenously released spores. They are produced from sporangiospores. So, sporangiophores, the bodies are what? Sporangiophores, from which sporangiospores are released out. This sporangiospores you can see it in Ascomycetes again. Ascomycetes. Fine, right? Here you see sexual mode of reproduction. Sexual reproduction is through oospores, ascospores and basidiospores. Oospores are non-motile and endogenously produced. Okay, non-motile endogenously produced. This you will see in oomycetes. Fine. Ascospores are endogenously released sexual spores observed in Ascomycetes. Basidiospores are the only exogenously released sexual spores which are non-motile, again observed in the class Basidiomycetes. Very important to remember the spores with respect to their classes, highly important. If you want, just take a screenshot and memorize it. Definitely you will get a question from this area. Okay, I will just give you 2 minutes of time, copy it down. Next you see here, based on the morphology of mycelium, mode of spore formation and fruiting bodies, Kingdom fungi is divided. So what is the basis of classification is again very important. Basis of fungal classification. Basis of fungal classification. So kingdom fungi here, if you see, is classified into 4 classes: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Phycomycetes, you have 2 subclasses in it, Oomycetes and Zygomycetes. Okay, 2 subclasses under Phycomycetes are what? Oomycetes and Zygomycetes. What is the basis of classification over here? Morphology of mycelium, mode of spore formation and fruiting bodies. Here if you see Phycomycetes, the general characteristics of Phycomycetes include, it is also called as what? Algal fungi. They are found in aquatic habitats on decaying wood and they act as what? Obligate parasites, and they have primitive mycelium like aseptate mycelium. Asexual reproduction is through zoospores, aplanospores and sporangiospores as well. Sporangiospores. Asexual spores and sexual spores of each class of fungi are important, ma. And here sexual reproduction includes fusion of 2 gametes. Either there are 3 modes of sexual reproduction. Fusion of gametes can occur through isogamy, anisogamy and oogamy. Isogamy, when 2 gametes are similar in morphology and physiology. Anisogamy, either there is morphological differentiation or physiological differentiation in between the gametes, then it is said as anisogamy. If small motile male gamete is fusing with large non-motile female gamete, is called as what? Oogamy. Okay, isogamy, anisogamy and oogamy. Hope this is clear to all of you. Next, products of the gametic fusion through any of these modes. Product of the gametic fusion is oospore in Oomycetes and zygospore in the Zygomycetes. These are the products of sexual reproduction or gametic fusion. Oospore, you can see it in Oomycetes, zygospore in Zygomycetes. These are again the sites of what? Meiosis. Post meiosis, haploid spores are produced and which develops into what? New hyphae. So here you can see zygospore, which is formed after fusion, gametic fusion. And this is after meiosis, it is producing new haploid spores which develops into a new hyphae. Hope this is clear, right ma, for Phycomycetes. Examples if you see. Examples if you see, we will stop here ma till Phycomycetes. Examples if you see, for example, mucor, rhizopus is also called as what? Bread mold and albugo. This you can memorize with a mnemonic like RAM, R-A-M. Rhizopus, which is also called as bread mold, albugo and mucor, which gives white spots on mustard leaves. Okay. So, we will enclose the session till here, and in our next class we will finish the fungi and rest of the chapter, and we will also exercise the EAPCET question. So, whoever are attending for the next session, revise the concepts till here and comma, because it will be helpful for you to solve the EAPCET question in live. Okay. So, that is all for today's session, students. Thank you.